ovarian stem cells should we be considering and preparing for clinical application? And we will have two presentations in this context, one against and one for.
Okay, well, I'd like to thank David for that excellent introduction. But the topic that I saw that we were defining was in terms of the stem cells that are derived from the ovary. So I'm going to focus on these ovarian stem cells. Should we be considering, so we're not saying, is it time for clinical application? We're saying, should we be considering engaging in a debate, engaging in what is required to get them to grow and develop, and whether it is something that would have any utility? Should we be considering and preparing for clinical application? And I'm taking the position that yes, we should. So here's my declaration. When I was asked to do this talk, I had no declaration, but recently I've been awarded a research contract from Ovacymes, and we are testing some of their cells. So if we look at the ovary and the development of the ovary, of course, the majority of the follicles, the oocytes, are within primordial follicles. They're the resting follicles. And these follicles in the human are formed before birth. And then they're resting and they're activated to grow throughout life. So in an older woman, these oocytes have been hanging around for a long time. And it has been a debate for a number of years whether the ovary, mammalian ovary, has a capacity for regeneration. Can they form new eggs? Now, not all mammals are the same. We know that in other species that there is regeneration, and we know that in some mammals, such as the prosimian primates, lemurs, there's certainly the cells there for the capacity for regeneration. They seem to form new oogonia. But, of course, no one's really studied them in great detail. And our models all come from mouse, has mainly come from the mouse. But recently, so prior to this, and David mentioned in 2004, there was a sort of reigniting of the debate as to whether the mammalian ovary had the capacity for regeneration. And that was the work of Joshua Johnson and John Tilley, at that time in Mass General Hospital. And they proposed the idea, based on counting studies, that the ovary looked like it was making new oocytes. Now, this was going against a dogma, absolutely a dogma that we'd believed since the 1950s, the work of Solly Zuckerman in Birmingham, that there was no regeneration in the mammalian ovary. And then there was work on mouse, and of course there's been people that can't reproduce this and so on. And of course we now have the work on induced pluripotent stem cells and so on, which I don't think anyone else has reproduced in the Hayashi and Sato work. It's elegant work, but clearly this type of work is incredibly difficult to do. It's not every lab that's going to be capable of doing it. So Tilley's group isolated cells, not just from mouse. Same time, there was a group in China isolating cells from mouse that seemed to have the potential to form new oocytes. But in 2012, they isolated a population of cells from the human ovary. And these cells were characterized by having DDX4, so VASA. So a germ cell marker, and we can argue about whether it's a germline marker or not, but it was accepted at that time there was a germline marker. And these cells could undergo proliferation. So that was a cell population that was isolated from by John Tilley's group. And that seemed to be quite an advance from the mouse studies. And what they did was they took these cells, they transfected them with the green fluorescent protein, they injected them into human cortical strips, and they found that these cells were forming what appeared to be new oocytes within primordial follicles. So that was quite an exciting discovery, that these cells were there within adult human ovaries. Now, recently there's been a paper published from the Chinese group, Jie Wu, in Shanghai, and they've also been able to produce what appeared to be germinal vesicle oocytes from cells that they've isolated, actually from aspirates. So they've got like small pieces of tissue through the ovary, and they've isolated a similar population of cells to Tilly. But these cells have created a huge amount of controversy. And there's a feeling that actually this is in some way, these cells don't really exist and they're not going to stand up to scrutiny. And that's the kind of background that we work on. Now, I know that my learner's friend David likes Stephen Jay Gould, and he had this quote yesterday. And it's a very good quote in the sense that when we really, really believe something, often we don't really want to see any new evidence appear. And I stand here and tell you that's how I felt when the work was being published by John Tilley's group. And I said, no, absolutely not. There is not a cell population in the adult ovary that is capable of forming new oocytes. So I was incredibly sceptical about this work. And we published critiques of Tilley's work. But of course, maybe being too dogmatic is not a good thing. We have to open up and then start to look at the evidence. So what I'm going to tell you about is a bit of my journey, and apologies to Mark Twain, a skeptic's progress. The question is, well, you know, what do these cells actually do? And I'm going to tell you how I became convinced that there is a population of cells within the ovary and that they appear to be capable of forming new oocytes. And that sort of change came about when John Tilley offered to give me some of the cells that he had isolated. And these cells, he asked if I would put them into the culture system that I spoke to you about earlier. And we injected these cells, so the green fluorescent protein cells, within the human ovarian cortical strips. And what we saw was, I mean, I honestly didn't think anything would happen, but what we saw was that the cells we injected in, that's the sort of asterisks, the stars here, they started to become larger structures. And we could see these quite clearly within the pieces of tissue. And we really didn't understand I didn't understand what was going on. So we then looked at the histology, and we could see these GFP cells in the tissue becoming larger and associating with somatic cells, and that's absolutely essential, and I think David has illustrated that really well, that a germ cell will not become a functional oocyte unless it has appropriate somatic cell support. And then we could see these GFP positive oocytes. I mean, if we look at the morphology, and anyone who looks at oocytes, I think would agree that it looks like an oocyte. Now, of course, looking like an oocyte doesn't necessarily mean that it's a functional oocyte. But this was quite an astounding piece of evidence for us. And we then thought, well, you know, there's something in this. There's cells within the ovary, and within the lab, we felt that we should look at these cells in more detail. So that led us into this whole area of the germline stem cells that are within the ovary. Now, we call them germline stem cells. We might say so-called or putative germline stem cells. The reality is we don't know their full characteristics. So whether we would call them a unipotent cell or a pluripotent, they don't seem to be pluripotent when we put them into the ovarian tissue. They seem to all form what appear to be oocytes and not the somatic line at all. So we could see these cells forming what appeared to be oocytes within that tissue. And that was really encouraging for us. And again, as someone who had been incredibly sceptical about these cells, it was really a sort of, you know, a part of a sort of wake-up call that, yeah, there's things in the ovary that we don't know about, and we should study them in more detail. But still, this work, of course, came in for a lot of scrutiny, and a lot of people, there was several groups that have tried to replicate, and not entirely replicate, but tried to obtain the cells that John Tilley had previously described. And most of the work was mainly in mouse, and many of the groups have been unable to do that. So when we set out within our lab, it was with trepidation in terms of what would we find within these adult ovaries. Would we be able to get the cells that Tilly had previously isolated? And the answer is that it was very difficult to do. We had to learn a lot that we didn't find out from the published work, and we had to modify things, particularly in terms of the dissociation and so on. But we essentially used the same protocol or the same basis for isolating these cells. And the basis of this was to use an antibody to DDX4, because the theory was that these cells would have DDX4 on the outer part of the cells, and they could be pulled down by FACS sorting, and we could then sort these cells and then look at their characteristics. So that's essentially what we did. We used a FACS-based protocol, and we looked for a small population of these small in size cells, and then looked to see what their characteristics were. So what we've done is most of the work that was published looked at these cells after they had been cultured, because they needed to get more material in order to carry out the PCR. But we were able to get enough material in order to analyse the cells immediately after collection. So it wasn't an in vitro transformation that we were looking at. We were looking at the cells that we freshly isolated and what was their profile then. And what we found was that it's not a homogeneous population. There's a heterogeneity in the population of cells that we pull down. And we know that there's probably a range of cells. Whether they're all going to do the same thing, we don't know, or whether they're at different stages of differentiation, again, we don't know. But we do see that they have germline markers, stem cell markers, and they don't have follicle or oocyte markers. So they're a different population of cells. And some of the cells or the populations that we pull out, and the difference in the populations is based on their intensity for DDX4 when we're doing the FACS sorting. And we find that some express dazzle and others don't. So we're now looking at these subpopulations and seeing whether they have different potentials or developmental potentials. But we were quite, again, surprised that we were able to get these populations. Now, there was a lot of, or there still is, an ongoing debate about whether DDX4 is really the basis by which these cells are pulled down. And do these cells have DDX4 in them? And we clearly showed that the positive cells have DDX4 when we look at the protein. We see DDX4 in the positive cells, and we don't see them in the negative cells. So we're getting two separate populations, and within the positive, there's probably subpopulations there. And we've done this with different antibodies. So the other thing too is, and which was very, very surprising to us, no one seemed to have looked at immunocytochemistry of the tissue, because you would think if the cells are there and you're pulling them down using this antibody, then look at the tissue. And this is a laborious process, and it's a postdoc in the lab, Marie McLaughlin, who did this work, where she did the immunocytochemistry of her DDX4 in human ovaries, and then went right through the sections and looked for other cells that came up positive for DDX4. And of course, the oocyte stain for DDX4 are controls here, skeletal muscle. There's no DDX4. And then when we look at the sections, you go through them, sift through all of them, you see these cells throughout the tissue. It's mainly in the cortex, but throughout the cortex, you'll see these positively stained DDX4 cells. What are those cells? These are cells that were never, ever explained before or even reported before. But we were seeing them consistently in almost all the ovarian material that we looked at. So we've isolated a population of cells that appear to have the characteristics of a germline cell, and we're now testing them. We've isolated them in human, range of ages, and isolated them in the cow. And we've done some chimeric models in the cow where we've combined these cells, and this is a student, a PhD student with Richard Anderson and myself, Kelsey Grieve, in collaboration with Susanna Williams in Oxford, took these bovine ogonial stem cells that were marked and then combined with mouse somatic cells, so making a chimera. And when you have the somatic cells by themselves, you just see somatic cells. But when you combine them with the bovine putative ogonial stem cells, we can see follicles forming. So our conclusion is that these cells, if they're in the correct environment, can form what appear to be oocytes. Now, of course, the real test is whether they can be fertilised and form embryos. And, of course, that has not been done in our system yet. So, getting back to what David was saying, of course, then, making oocytes from either induced pluripotent stem cells or embryonic stem cells or these ogonial stem cells or germline stem cells is dependent upon the correct somatic cell support. Absolutely essential. And that's maybe where we don't entirely understand that. But it would seem with these human cells that we don't have to go back as early as in the mouse model. And we do have access. Richard Anderson has human fetal cells, and that, of course, is something that we're looking at in terms of combining them. And some of our students have been working on combining those somatic cells with these putative germline stem cells from the human. So this is a complex process. There's absolutely no doubt about it. But progress is being made. And if you speak to some clinicians who have read the papers from the Japanese group, there's an enormous sort of feeling of excitement. And the excitement is such that they're saying, we don't even have to start with primordial follicles. We can start with skin cells. And so that's why I'm arguing today that we should be at least considering what the potential clinical applications are and what's the best way to, pursue this work. So to engage in a dialogue rather than us all going off in different directions, to really understand, well, what would be the best source of cells and what's the potential of each of the, the studies that are being reported sporadically. So we can find a distinct population of cells with germ and stem cell markers. They're isolated from adult ovaries and prepubertal girls, and also in Turner's patients as well. It's a useful model for germ cell development. There's no question about that. Here we have something from a very early stage, and we can see them forming what appear to be oocytes, and we've got an opportunity to test whether these are normal or not. There are, of course, potential clinical applications, so we shouldn't ignore these clinical applications. We should engage with them and start to get a consensus and a debate rather than just simply ignoring the possibilities of these cells. So, of course, these could provide a source of gametes for research that would actually lead to improvements in fertility treatments. We need to define the clinical need for in vitro derived oocytes. David alluded to some of the conditions that might benefit from this, but I think it really needs a dialogue with the clinicians, with practitioners, to actually define what that need is. And also the potential of the technology. Could it be an alternative to egg donation? These are things that should be discussed now rather than waiting to the point where it becomes a reality and someone goes off and actually does it. There should be an engagement at this stage. We have to consider when you're storing tissue from patients, is it right that you store the tissue with the possibility that you might get these cells in the future and that they might form oocytes? We have an obligation to consider what the possibilities might be. So I believe that progress will only be made by engaging the clinical and scientific colleagues to come to a consensus and really put down a roadmap in terms of how we should progress with this type of work. So some people might say, okay, this is false hope. Is it false hope or is it good planning? And that's what we really have to think about. If we consider the field of fertility preservation, When Roger Gosden and David Baird cryopreserved the sheep ovary, they didn't know if that would work in humans. But Edinburgh had the foresight to develop a fertility preservation clinic, which Hamish Wallace and Richard Anderson now organise. And they were taking tissue and were using the methodology that Roger Gosden had developed for cryopreservation and storing those young women's tissue. Now, at that time, no one knew if that tissue would ever be used, if it would ever be functional. And we now know that that tissue can be thawed and transferred, and we now have, it's unclear exactly how many babies, but we now know there's probably over 100. And Edinburgh has had its first birth using that tissue that was stored some time ago. So there was forward planning. And I think with new techniques, we should be considering that. We have an obligation to do that. Now, IVF, the IVF revolution, in this area, there's always new things happening. But when it was developed, it was mired in controversy. And Bob Edwards started to think about IVF in Edinburgh when he was a PhD student in the 1950s. So it took that long to realise his idea of fertilising egg outside the body. And Bob Edwards was pilloried. He had a terrible time from the British establishment, from the clinical community, from the scientific community. He didn't get any research grants, really a terrible time. But a very humble man who was able to withstand that and move forward with, what has now been established, and all of us here, owe a debt to him. And when it was marked when Louise Brown was born, he said it marked the end of the beginning of the human IVF. And it was acclaimed by the Royal College of Obstetrics and Gynecology. And there were people even then saying, this is a fake. And of course, at the end, he said, they don't matter. It doesn't matter. But we have to, at some point, think, well, okay, what does matter? And how can we progress with these things? So we have to look at the future, and the field of the female germline stem cells is new. It's very new. We don't understand. We haven't even defined the cells to any great extent. The concentration of effort has been in doubting their existence. People will say, I don't believe in these cells. And it's not a religion. You're not being asked to have blind faith. It's to look at the evidence, look at it rationally. But it's time really to concentrate on defining the population of cells that reside within the mature ovary and determine what their utility may well be. And looking at some of the strategies that we can apply to cell-based therapies to treat infertility and, of course, fertility preservation. So what I would say is that we should be considering these. Techniques, technologies, they move quickly, and we're always gaining new knowledge, but we need to consider what the applications might be. They may come to nothing, but they may come to something. And so at that, if we agree on a consensus and we discuss it, then we're ready. And I'm sure Bob Edwards would have wished that IVF had that opportunity all those years ago. So in terms of the dogma about the germline stem cells, it has got mixed up. It's got mixed up with the dogma in terms of forming new eggs throughout life. And there's no evidence that the mammalian ovary or the human ovary forms new eggs under normal conditions. But the fact that there's cells there that may have the capacity to form new eggs under the conditions that we create, then we should definitely look at it. So we have to sort of move away from the dogmatic view and sort of be more open about new techniques and new technologies. And of course, that's the thing that's made IVF and made the whole assisted reproduction technologies what they are. So I would acknowledge then the people who have been involved in this work, and of course, and I'd acknowledge David Albertini for participating in this discussion.